to the BBS research seminar for this. Speaker will be given by Rebecca Sleater, who works as a environmental specialist. So if you are interested in something to do with a career open package, that would be a good thing to do. This is Dr. Erin Vogel. She is currently at Rutgers University, where she was a professor in the Department of Ecology. She started her career at Colby College in Maine, and when she finished there, she went on to do a PhD at Stony Brook, where I met her for the first time. At Stony Brook, she did research in capuchin monkeys in Costa Rica, and then followed that up with a postdoc at Santa Cruz, a postdoc at the University of Zurich, another postdoc at George Washington University, and from there she went to Rutgers, where she is currently working. So please join me in welcoming her.
Thank you. Okay. Excited to be here today. Some ecologists and biologists. I don't know. My history is that my PhD is actually in ecology and evolution, but because I study non-human primates, we tend to get funneled into anthropology programs, and we have to kind of justify why our research is relevant to human evolution, which I think I've done a pretty good job of doing, but I won't be talking about that with you all today. Instead, I'm going to talk about some of the research that's been coming out of my lab for the last 5 years. On the work I've been doing on wild orangutans in Indonesia. And oh, hold on, sorry. And before we started, I just want to thank project where we collect the data year-round. We have a full staff that are collecting behavioral data year-round, and a lot of students that have contributed to our long-term database and all sample collections. And the reason I went to Southeast Asia in the first place after doing my PhD on capuchin monkey nutrition and ecology was because I was really fascinated by the rainforests in Southeast Asia. And these rainforests are really characterized by unpredictable and really big fluctuations in the availability of fruit resources. So the phenology patterns are really different than other tropical forests because they're unpredictable. I work over the past 20 years or so. By masting, which means that everything fruits at once, and then you have these long periods of like 3 to 7 years where there's basically no fruit available. The forest where I work is not a masting forest, and I'm going to talk about what we're finding in this non-masting forest, but there's still a unpredictability in fruiting and in the amount of fruit available. So if you are a very large arboreal frugivore, in fact, the largest arboreal frugivore in the world, you really have to figure out how you're going to survive, what you're going to do during these episodes of extreme frugivore. Some of them can be really long. So for example, in October of 2015, we had a really major fire period in Southeast Asia, and it was followed by about 36 months of extremely low fruit availability. So you can imagine if you need to maintain this bond, which is in Uganda, chimps eat about 111 species at this site and about 179 unique species parts. And if we look at orangutans, they're feeding on over 230 different species, many of which we still don't know. So there's about 60 unknown in addition to that. And we bring in botanists quite often to try to ID these species, but over 107 unique species and parts. So for one species, they may feed on 4 different parts: bark, mature leaves, immature leaves, ripe fruit, shoots, all different types of resources items from the same tree. When fruit is available, prefer to feed on fruit, right? So you can see that during these episodes of really high fruit availability, about 75% other items in their diet. So they do what we call this dietary behavioral switching. And this is one way they're able to cope with these episodes of scarcity. In the late 1990s, my colleague Cheryl Knott, who's at Boston University, for her dissertation research, what she found is that during these periods of low fruit availability, she was collecting their urine samples, and she noticed that during episodes of fruit scarcity and mass force, these orangutans are actually burning body fat. And with evidence, she was able to determine this by looking at evidence of ketone bodies in their urine. Ketone bodies are a byproduct of fat metabolism. So it's like a really popular diet over the past couple of years where you eat a lot. Fat. And so Dr. Knott proposed that during these high fruit periods, they actually build up these fat reserves and then they burn them during the low periods. And among non-human primates, this pattern seems to be pretty unique. For example, my colleague, Melissa Emery Thompson, has collected over 15,000 chimpanzee urine samples from the population that she studies, and only about 0.06% have evidence of ketone bodies, where for us, it's somewhere around 10 to 12%, depending on the site. Other primate species, they just don't protect them. So the question is, why is that? Are they not burning body fat, or do they not have enough body fat, or do they maintain kind of neutral energy balance a lot of the time, be able to? And that's really what they think of these chimpanzees, is that their habitat is a lot of fruit, so they're not. Total energy expenditure, I should say, of orangutans, Pongo, gorillas, chimpanzees, and humans. And we found that for a given fat-free body mass, orangutans just have a slower energy expenditure like the water method. And what that means is that on a per cellular basis, their metabolism is basically slowed down compared to other great apes, including ourselves. They've just figured out a way to slow everything down. And if they looked at resting metabolic rate calculated the percent expected metabolic rate, they found again suggested that this is potentially an with these unpredictable and long episodes. Is that orangutans are most likely more efficient at getting energy from protein sources relative to other primates that they looked at, including humans, chimps, macaques. Oh, and then they also had an outgroup there. So another potential adaptation for coping and maximizing the energy they can obtain from non-carbohydrate energy sources, things other than fruit, for example. So a little more and think about what are the nutritional strategies that primates help with unpredictability. And one thing that has been proposed is what's called the geometric framework of nutrition. And that maybe it's not all about maximizing energy intake, those optimality models suggest, but it's about the balance of these nutrients. In the face of this natural variation in food types and macronutrient availability. And I'm mostly going to talk about the primate work because this is really relevant to what we're doing. But basically what studies have found is that more frugivorous primate species like the spider monkey tend to maintain a relatively constant level of protein intake with a lot of variation in non-protein energy. So things like fat and carbohydrates and fiber. And that's a pattern we see with frugivores. So they call it a pattern of protein prioritization. I don't really like that terminology because it sounds like they're going out and just prioritizing eating. I don't think that's what they're doing. It maintains a very tight amount of protein consumer basis is very narrow, and that's the range is very narrow. And that's the same thing we see in humans. Gorillas, on the other hand, that are more proliferous, is about the same on a daily basis. And then macaques and shifacas do kind of a mix of all different types of things where they're maintaining the same ratio or they just decrease the total amount of macronutrients depending on the season. And Jessica Rothman, who's one of my collaborators on several projects that we have going on right now, has looked at this in a number of different non-human primate species. And and what to use this, I guess. And what she's found is that the frugivores tend to have a higher ratio of non-protein energy to protein, and then the more species, including humans, tend to have a little bit less non-protein energy to protein, and then the folivorous primates tend to have more protein relative to non-protein energy. Started to ask questions about their behavioral and physiological adaptations that really enable them to tolerate this unpredictable and variable amount of food in their environment. And so if we just look at food items, on this axis is energy from fat, energy from protein going this way, and then energy from carbohydrates. And you can see that there's a lot of variation in energy from fat, but not as much variation in energy from protein. Most foods are 5% protein, although some of them are higher, especially like insects. And then in terms of carbohydrates, most of their foods are pretty high in carbohydrates, but not all of them. So we started thinking of looking into the literature about metabolic stability. the fuel, depending on what's available. And it really has become an important framework for understanding what are the adaptive capacities that species have, especially when they're faced with extreme environmental variation. So today I'm going to talk about how this unique ecology of Southeast Asia influences the dietary strategy of orangutans, which I've kind of already talked about, and then the physiological adaptations that we see. Further, I'm going to talk a little bit at the end about how this influences their health. So we've made several predictions for this study. This has been an ongoing study for 6 years. About this, finally, some paper. Yeah, I will eventually. But the first prediction was that food availability will low. We expect to see an increase in the mobilization of fat, so increased ketosis, and a decrease in what are called urinary C-peptides, which are linked to insulin production in a 1-to-1 ratio. We then predicted also that during these periods of low caloric or carbohydrate intake, that orangutans would use a gluconeogenic pathway, and they would actually start using protein more often for energy, and even their body protein for energy. So skeletal muscle wasting, which is something that we observed previously in another study. And I'm going to talk about that a little more. Finally, the most important thing to think about is, are these episodes of scarcity really that tough? And maybe because of these unique adaptations that orangutans have, maybe they're not that tough. In fact, maybe they're beneficial. So I'm going to talk about some work that an undergrad in my lab has been doing. He's now actually a graduate student, but not in my lab, but he's been working on this question. So all of the data I'm going to be presenting to you today were collected at the Tuanan Orangutan Research Station in Central Kalimantan, which is on the island of Borneo. So this is the island of Borneo. This is the Indonesian part of Kalimantan, and this is the Tuanan Orangutan Research Station. It's right along the Kapuas River, and it's in the west part of the Mawas Conservation Area, which is a 360,000 hectare reserve of mixed usage types. We've been studying about 134 fully habituated animals. That means we hope that their behavior doesn't change when we're present, although our camera trap data seems to suggest that they're on the ground more than we originally thought. So maybe we are affecting them a little bit. We do nest-to-nest focal follow. So we follow them from the time they come out of their nest to the time they build a new nest, and they make a new nest every evening in the trees. So usually it's about 12 to 14 hours of following them during the day. And we also have about 2,500 trees that are tagged in our phenology plots that we've been monitoring every month for the last 20 years. So since 2003, we set up the phenology plots, and we've expanded them. We expand them every year, basically, as new trees come into the plot. So new trees as in those that are greater than 10 centimeters DBH. I'm also going to talk about these urinary biomarkers, and a lot of people ask me, do we actually collect their urine? It's not as hard as I think. When they come out of their nests first thing in the morning, they urinate, just like when you wake up in the morning, you urinate, right? They do the same thing. So they come out of their nest and they pee, and we make a little tool like this with a stick and a little bag at the end that we loop around the edge of the stick, and we just wait and we wait. And then when they come out of the nest at like 5:30 in the morning, they always pee, and we catch their pee, and then we pipette it into, we test it for ketone bodies and other markers using chem strips that your doctors use in their offices. They're just urinary test sticks. And then we bottle it up and freeze it in our solar freezers. And eventually those samples make it to my lab at Rutgers for analyses. And today I'll talk about some of the things that we do in the lab, like C-peptides of insulin using radioimmunoassays. This is a great marker of energy balance. We look at urea concentration using a colorimetric reaction in my lab. That's a really good method to look at either exogenous protein, excess exogenous protein, or the use of body protein via gluconeogenesis. And we also do nitrogen isotopes, not in my lab, but we prep them in the lab and then we send them out for analysis at my colleague's lab. And delta N15 is a really good indicator of if the orangutans are basically eating their own meat, so they would go up a trophic level. And because orangutans don't eat meat, well, they rarely eat meat. I shouldn't say they don't. They rarely eat meat. This is really indicative of skeletal muscle wasting in this species. I have a wonderful former graduate student who is a statistical whiz, which has been really great for me. He's currently at Max Planck, but he ran all of the analyses I'll be talking about today. We used Bayesian generalized additive mixed models with a 95% credibility threshold. And in these models, we included a lot of different variables, including sex, fruit availability, time of the sample collection, and we include individual ID. So the ID of the individual has a random effect in all models. And one of the things you'll be seeing a lot of figures that look like this. What Shaheen was able to do is we were really interested in understanding with Bayesian statistics, obviously, you don't have like a significance value. So what we were interested in were the regions of the curve where they had a non-zero rate of change. So to do this, Shaheen used the method of finite differences to estimate the first derivatives. That gives us the rate of change, and then we could test to see if that rate of change is different than zero. So where you see these blue lines, that means that the blue sections of the curve means that they're significantly different than zero, the rate of change. This is a graph of fruit availability again at the site. Average fruit availability is just below 5% of fruiting trees. So you'll see a bunch of graphs like this. And I point this out because I think it's important. We seem to see a lot of effects when fruit availability is either below the average of the habitat or above the average of the habitat. So I'll point that out in some of these result slides as well. So the first question was, do these orangutans use a strategy of protein prioritization? And just looking at the graph, it's pretty clear that they do. The ratio of non-protein to protein is about 10-1. This is about the same that we see in spider monkeys and other really frugivorous primates. One thing that's really interesting is that we really don't see a lot of change in available protein intake. We see about a 3% difference between the low to the high fruit period, or 4% difference. Whereas if you look at non-protein energy or the ratio of non-protein energy to protein, the percent change is much greater. So there really have a lot of variation in non-protein energy, so fats, non-structural carbohydrates, structural carbohydrates, but very little variation in protein intake. But we do see an effect of fruit availability on the ratio. So if we look at non-protein energy to during the high fruit period compared to the low fruit period, it decreases from 11.6 to 8.5. So they are changing their strategy a little bit and consuming a little more protein or less, or maybe they maintain the same amount of protein, but are just consuming a lot less carbohydrates. That's really what's going on. And if we look at the relationship between the fruit availability index and these different variables, what we see is that there is a little portion of the curve that was significant, but overall it's pretty flat, and the amount of explained variance was very low for protein. Whereas for non-protein energy and the ratio of non-protein energy to protein, right about the average of FAI is where we see this relationship kind of flatten out. And before, when fruit availability is below average, is where we see a change in non-protein energy intake and the ratio. I'm just going to go through some of these results. We've actually found no relationship at all, no matter how many analyses we did or how we looked at the data. We found no relationship between urinary C-peptides of insulin and the availability of fruit, or urinary C-peptides with macronutrient intake, caloric intake, or anything. And I was really stumped by this because previous studies on orangutans have found relationship, like when fruit availability was really high or caloric intake was really high, they had higher levels of urinary C-peptides. we need to really explore a lot more, because the thing I'm thinking about is that urinary CPR is a really good indicator. We're looking at energy intake. So it's very likely, and we do know that during low fruit periods when caloric intake is reduced, the orangutans move less. So maybe they reduce their energy expenditure during these low fruit periods and compensate, so we don't see an effect on insulin production. Or it could just be that they're really not as sensitive to insulin, but we don't know. Interestingly, we also did not find a relationship between ketone body presence and FA and fruit availability, and we found very few relationships with the other macros. But what we did find is that there was a weak, very, very, very weak relationship with the ratio of lipids to total non-structural carbohydrates. However, I do want to mention that this is on a scale of 0 to 10. I mean, 0 to 1. It's either the presence or absence of ketone bodies. And you can see that the significant portion of the curve is like, we're talking about a change from 0.03% to 0.05%. So it's really insignificant. So what's going on? This slide should have been earlier, but basically, this is the relationship between these different markers of dietary, of energetic status and fruit availability. And urea is the only one where we see a very strong relationship with fruit availability, where when fruit is really low, we see really high levels of urea concentration, but everything else was pretty flat. Now, what's going on with this, with the ketone bodies? Well, this is days of a follow set. So we usually follow one animal for 10 days straight. Sometimes we don't get to follow them for 10 days straight because they like lose us. They're very good at losing us. They like to lose us. They don't want us there. So we try to stay with them for 10 days, but you can see that some of these are much shorter. Some of them are 5 days. And sometimes we switch animals too. So if they come together, they're very more solitary than other apes. So when they do come together and they have what we call a party, sometimes we'll switch focal animals. We'll be like, oh, we follow this person, this individual last year, so we'll just go with somebody else that we haven't file and get data on. So after 5 days, we often will switch if we find someone new. Yeah, we call, like, when they're within 50 meters of each other, a social party. So they're really not very social animals. It's usually just a mom and her offspring. Unlike other non-human primates that are in these big social groups, orangutans are like really not as social. We'll just put it that way. But what you can see is that on a daily basis, they go in and out of ketosis quite often. So one of the things we're thinking is that, and you know, these are samples where there's no ketone body. Sometimes we'll find one. Sometimes we'll find a spike and then it goes back down. So what we're thinking is that maybe we're missing things because we don't sample their urine every day. So it might be that they're compensating and going in and out of ketosis, and we might just be missing some of that variation in our sampling. What we'd like to do next is we've just validated a quantitative ELISA assay in my lab for looking at beta hydroxybutyrate, which is one of the components of ketone bodies. So we're going to move on to doing that and see if we can see a more precise measurement of ketone bodies. But what was really cool is when we looked at urea concentrations and nutrient intake. So remember, urea lets us know if they're over consuming protein. So if you have a really high urea concentration, that either means that you're over consuming protein and you're just getting rid of the nitrogen waste, or potentially that you're utilizing a different pathway to get your energy through gluconeogenesis. So a little more costly pathway. And what we found is that typically when non-protein energy, the ratio of non-protein energy, carbohydrates, calories, when any of these things are really low, we're seeing urea concentration is higher. So when these individuals are taking in less calories, when they're taking in less carbohydrates, when they're taking in less fat, we're seeing that urea concentration is increasing. What's really interesting, though, is that we don't see an effect of protein intake, because protein intake is pretty stable all the time, but we're not seeing a relationship between urea and protein intake. So this suggests that it's not all about diet. It's not all about protein intake and using protein for energy. It's about using other non-carbohydrate sources for energy as well, including their own body tissues. That just highlights that all the relationships that were somewhat significant. We also looked at delta N15, which, remember, is the nitrogen isotopes, which gives us an idea of skeletal muscle catabolism. And we did find a relationship in delta N15 between total non-structural carbohydrates and non-protein energy and total caloric intake, but just at the really low levels. So when they're taking in really low levels of these things, that's when we start to see skeletal muscle catabolism. And to demonstrate this, you can see that below about 3,800 kilocalories per day, it looks like they're starting to use exogenous non-carbohydrate energy sources for energy and endogenous lipids, so their body fat. And then when they're taking in below about 800 kilocalories a day is when we start to see them start utilizing their skeletal muscle. And one of my postdocs, Caitlin O'Connell, about a year ago, did some more analyses in the lab, and we used this method looking at the residuals of creatinine to specific gravity. And specific gravity is about the density, basically, of the urine. It's just relative to water. What is the density of the urine sample? And then we measured creatinine in the lab, which is produced pretty much at a constant rate, but it gives you an idea of muscle. It varies with muscle, the amount of muscle that an individual has. So what we were able to do is use this method to estimate skeletal muscle. And what we did is we looked at high fruit periods and low fruit periods across adult males, both unflanged and flanged males. There's two adult morphologies for males. They look very different. And then there's adult females, and we looked at adolescents independent, so kids that are still nursing. And what we found is across the board, all individuals have reduced skeletal muscle during these low fruit periods. So it was very clear to us based on this study that they really are using their skeletal muscle for energy during these low fruit periods. So we think that what's going on is that, these orangutans are really adapted to these low fruit periods, and that they show a lot of adaptations for efficient energy acquisition during low fruit periods. They have reduced energy expenditure, according to Ponser's work. They have low resting metabolism. And our work has also showed that during these low fruit periods, they really reduce their activity levels. They just kind of sit around and eat leaves all day and sleep a lot. So males typically during low fruit period, the adult males will go to bed, will make a nest at like 2 or 3 in the afternoon. Females maybe a little later, like 4 or 5. Because they're still lactating and have to make sure they can feed their kids. We also are seeing what's suggestive of gluconeogenesis of amino acids. And we think what's going on here is that this is providing enough glucose to supplement their energy metabolism. So it's maintaining their insulin secretion. And maybe that's accounting also for this lack of urinary C-peptides that we see with reduced caloric intake. And perhaps it's beneficial for them to break down that skeletal muscle during the periods of low fruit availability and build it back up during these periods of high fruit availability, because they don't have to, it's costly actually to maintain this metabolically active muscle tissue. So maybe this is the strategy they have during these low fruit periods that are really unpredictable, to start breaking that down for energy, and then they can just build it back up when they have their boom period. So I'm going to end this talk with talking about, are these episodes of caloric restriction actually harmful? Because this is something we've always thought as people that study orangutans, like, oh, they're struggling through these low fruit periods. They're really harmful for them. Yet orangutans do really well in the wild. And they have one of the highest survivalships out of any mammalian species, except for like Swiss people in Europe or some, it's really incredible to see how well they do. And their infant survivorship is also extremely high. So Daniel Nemenko was an undergrad in my lab, and he did a senior thesis with me. And then he's finishing up his PhD right now at University of Colorado Boulder, doing work on humans, but we'll excuse him for that. And he started studying this for his senior thesis and has come back to the lab every summer to do more analyses, and we've worked on them together. And one of he's looking at is oxidative, thinking about if during these episodes of caloric intake, do we see patterns with oxidative stress and oxidative damage? So very briefly, reactive oxygen species, for reactive oxygen species, there are both endogenous and exogenous sources. So internal sources of reactive oxygen species and external. And really, you're producing reactive oxygen species all the time, right? They're a byproduct of cellular metabolism in the mitochondria, but they can really damage if they build up proteins, lipid membranes, DNA, and they're really tightly linked to biological aging. So of course, our body has ways to counter them by producing antioxidants. So antioxidants then are produced by our body, or we can get them in diet. There's different sources. There's endogenous and exogenous sources as well. And there are a lot of the exogenous sources of antioxidants can be through the diet, and it will influence them. But more recent studies have shown that actually the ones that we're producing all the time inside are also really beneficial. So there's still a question about how much diet is influencing our antioxidant production. But basically, when we have more reactive oxygen species than antioxidants, then you're in a state, or individuals are in a state of oxidative stress. If it goes the other way, where we have baseline antioxidants are upregulated relative to reactive oxygen species, then individuals are not in a state of oxidative stress. So this is just kind of an example again of what we can see when individuals are in a state of oxidative stress. We see oxidative damage. This can result in tissue injury. It's been shown to result in inflammation as well. And so I'm going to talk a little bit about that and some of the things we found. And overall, oxidative damage has been tightly linked to a number of chronic diseases and disorders in humans and other model mammal species. So how does this work? Well, a lot of studies have looked at the effects of caloric restriction on oxidative stress and health in different species. So for example, in mice, if we look at lifespan and caloric intake per week, individuals that were put on a high calorie diet had shorter lifespans. If we look at rhesus macaques, study in 2017, this is the percent of age-related morbidities. This is caloric restricted diet, and this is the control diet. You can see that individuals on a caloric restricted diet have fewer age-related morbidities. And then a more recent study on mouse lemurs showed that individuals on a caloric restricted diet survive more. Percent survival is higher. So we also know that this is true in humans. There's been several studies done on humans of these caloric restricted diets. Of course, you know, it's up to a point, right? But individuals that have a 25%, in this study at least, 25% reduction in calories also showed a decrease in one of the markers of oxidative stress that's found in the blood. So Daniel wanted to go out and figure out if we could do this with urine, since we had all this frozen orangutan urine in my freezer. And so we looked at that, and we looked at 8-OHCG using an ELISA technique in my lab. And we also looked at total antioxidant capacity, and we measured neopterin also, which is a measure of inflammation. Because many of these studies have found that when individuals have higher levels of oxidative damage, they also have higher levels of inflammation. And we all know that inflammation is just not good, right? It's an immune response. So real briefly, what did he find? When we looked at caloric intake, so this is caloric intake from the day before, which is what I showed you also in all those graphs before. When we look at previous day intakes relative to oxidative damage, what we see is that when they have a really high calorie diet, they have higher levels of oxidative damage, just like we see in humans, just like we see in other species. And we did find a very weak relationship, and you can see that by the amount of explained variance between C-peptides of insulin and oxidative damage. So when they had slightly higher levels of C-peptides of insulin, they also had higher levels of oxidative damage. So more insulin, more oxidative stress as well. We then looked at the relationship between 8-OHCG and total antioxidant capacity. And as expected, we found a really nice relationship between oxidative stress and antioxidants. So when individuals are experiencing more oxidative stress, they're also experiencing greater levels of antioxidants in their urine, or outputting. And it's about 54% of the variation in total antioxidant capacity was explained by our marker of oxidative stress. However, one thing we don't know that I will mention is whether or not the antioxidants that we're seeing is endogenous. So are they producing that internally, or is it coming from a change in their diet? So is it a counter kind of, are they counterbalancing these reactive oxygen species by eating foods that are high in antioxidants? And that's something that we're starting to study now in Indonesia. So we're doing these analyses in Indonesia on plant samples to see what foods are high in antioxidant production. But what was most exciting to me was the relationship between neopterin, which is our marker of inflammation, and 8-OHDG. So when they experience higher levels of neopterin, they're also experiencing higher levels of oxidative damage as well, which is what we had expected and what's been found in humans. So it kind of just validated that what we're measuring is we're seeing the right patterns that we would expect. So overall, in summary, we see that reduced caloric intake and this negative energy balance, we see a decrease in oxidative damage and a decrease in oxidative antioxidants. We also see a decrease in inflammation. So maybe we really do need to kind of rethink everything that we've been thinking about orangutans struggling through these episodes of scarcity. I mean, of course, like 36 months of scarcity or, 5 years of scarcity may be a little bit much, but, maybe these episodes of caloric restriction are not as harmful as we've proposed. And in fact, when we look at survivorship of orangutans, elephants, chimps, gorillas, and another population of chimpanzees, and even if we look at human populations, hunter-gatherer societies, which is right here, that bar graph there, what we see is that orangutans, actually, if we look at survivorship until the age of first birth, they're at about 94% survivorship to the age of their first birth. Whereas there's no other species that have been looked at that have that same level of survivorship, except for Swiss people in yellow. There have been other studies. So there was a really cool meta-analysis that was done, and each one of these dots represents a different bird species. And what they found is that species that live longer, that have higher maximum lifespan, have lower levels of oxidative stress using similar markers that we used, but they were using serum. So perhaps we need to kind of rethink what we've been saying all along, and these episodes of caloric restriction are beneficial and not harmful. And maybe this is why we don't see obese orangutans in the forest, right? You put orangutans in a zoo, or you put orangutans in a high caloric environment, they get really fat. And it's a problem when you talk to zookeepers, they always say, our orangutans are all overweight. They're all getting fat. And I say to them, well, it's because you're feeding them just all fruit. You need to give them vegetables. You need to give them broccoli. You need to, give them something that isn't pure sugar, because they're not used to getting that in the wild. They prefer it, just like we do, right? You put us in a high calorie environment, you walk into like a Quick Check, and you're just like, you're really hungry. You're going to like grab whatever you can and eat it. Orangutans will do the same thing. So we are adapted to really like sugar. And that's probably why we're experiencing a worldwide global obesity epidemic right now. In the forest, they experience these highs and lows constantly. So even when they build up some fat reserves, they're immediately going to burn them off. So we think that, based on these metabolic adaptations that they have for low energy throughput combined with their metabolic flexibility, their ability to move between different substrates quite easily for nutrition, they're actually doing okay. And orangutans aren't suffering through these periods of low fruit availability. So I want to thank you all for listening to me. I want to thank my entire amazing team in Indonesia and all of the students that have collected these data over the years, and my Indonesian colleagues as well, who make this study happen, and all of our funders along the way. So I can answer questions if anyone has a question.
If you do have a question, just wait a second for me to bring.
You can also have questions about like field work or anything.
in these periods of low fruit production, do they change the amount of time they spend foraging? Does that behavior also change?
They're constantly like chewing the food that they've foraged for. So I feel like processing time is longer as well. It probably does. So we know that gut transit times are quite variable depending on how much fiber and particle size in the diet. So it probably does alter their gut transit times as well. I would love to be able to do some kind of experiment where we label things, but it's so hard to know what they're going to eat. So.
Quick question. You had talked about the way you collect urine samples. Do they urinate only in the morning, or do they urinate throughout the day as well?
They urinate throughout the day, but hormones have cyclical patterns throughout the day. So we tend to try to collect the first void in the morning, but sometimes we do collect samples later on in the day too. It's just harder to get a sample because then you're following them around with this giant stick and they're like looking at you and what is this person doing? They already think we're crazy, probably picking up their pee and poop all the time. I don't know. Yeah.
During the low fruit availability, you said that they have like they're like kind of like moving around less. So then if they're eating more leaves during that time, like I would be thinking that they would like climb trees to get more leaves. So then how does that like relate to them having lower like sorry, like they're moving around less, but like I would think that they had to climb the trees more to get leaves for that variation diet.
Right. No, that's a great question. So during the low fruit, during the low fruit period, you would think they'd be actually traveling more because they have to get to those leaves, but because they're arboreal and there's a lot, leaves are kind of ubiquitous. Now they don't eat every species of, tree that's out there, the leaves of all different species, but the ones that they do target, they're pretty abundant in the forest. And there's certain species, there's particularly one species that one of my graduate students he's getting ready to submit a manuscript on this actually. There's one species that is a nitrogen fixing species as well. And it's very, very, very abundant in our forest. And orangutans, this is crazy. His study from our long-term database has showed that about 50 to 60% of their calories come from this one species, and they feed on the fruits, they feed on the seeds, and they feed on the leaves of this one species. It's the legume. It's a Polycarpus leucoconphus is its Latin name, and they feed on it all the time. It's a very, very important species at the site. The weird thing is there's a site that's like 70 kilometers away from our site where they don't feed on it at all. And it's present, but they barely feed on it. So but at our site, it's a very, very important staple food item. And it's given their diversity in diet, the fact that it's making up about 50 to 60% of their calories is crazy. So the point is that during the low fruit season, because they're feeding on leaves, they really don't have to move around that much. They just kind of are up there in the canopy and they're eating leaves and then they sleep. And then they eat more leaves, wake up. Sometimes they'll like take leaves and bring them into a nest. They'll just gather like a bunch of branches and just bring it in the nest and just sit in there and eat all night. I mean, it's pretty amazing. Are there any other questions? Yeah, no, you can ask. I actually, if you wanted to ask questions about what it's like to get into this kind of field work, or, how I, what my students have done, I'd be happy to answer any questions about the field station, about anything. Yeah.
I wanted to ask what made you interested in like orangutans in the first place?
That's a great question. So when I was my advisor studying primates, it was really interesting. As long as so I studied name is Carl. And he was really, we were talking one time at a conference, and he was talking about culture, and he was talking about how they learn their diets from their mom, and it's all cultural. And given that I came from such a strong ecology background, and my advisor was very quantitative and into the kind of optimality modeling, I said, well, how do you know they're just not eating the optimal diet? How do you know it's culture? How do you know they're learning? How do you know they just don't come across fruit and eat it because it's high in energy? So he's like, well, we need to test that. So that's kind of how I got orangutans. And I actually never thought I would stay there for studying them, but they're just really interesting because they have these crazy dietary adaptations. We don't that we've studied.
How does this work with your teaching schedule? Like, how often are you out in the field versus back at Rutgers and things like that?
So usually I try to stack my teaching in the spring, and I haven't been teaching as much in the fall. So I usually go to the field in November and part of December, and then I go for the summers and I teach in the spring. But that's been more recent. And I'm not in the field that long as well. Unfortunately, I wish I could stay longer, but I have kids and life and all those things. So I'm usually there for about a month and a half, from like 6 weeks in the fall, and then I go for like 6 to 8 weeks in the summer, and I can bring my kids to the summer school.
Actually, I wanted to ask about kind of your international collaboration. So you're working in a place where obviously you have a lot of Indonesian colleagues who are working with you. And in fact, I mean, Borneo has kind of 3 different basically states that are there, right? So mostly I was kind of interested in like what kind of Indonesian or institutional support do you get there? It's such a big project, how, and things like orangutans and palm oil come up a lot. So how involved is that? Are those institutions in conservation, including like the government, and how much they support this kind of work?
Yeah, they've been really wonderful. So I'm really fortunate. Carl Van Schaik worked with my colleagues, Tatang Mitra Setia and Suci Utami at MOCO, who are both at Universitas Nasional in Jakarta. He's worked with them since he was, doing work in Indonesia, 40 years ago. And so when I started as his postdoc, I really got to work with them and had the privilege of being able to do my research with them. And we just built up this project together. And I'm really fortunate because they're extremely supportive. I mean, we've trained over 150 students from Universitas Nasional, and students from local universities as well come. And the government's been really supportive. We also work with an organization called the Bornean Orangutan Survival Foundation. And they are an Indonesian NGO, but they also have NGOs all over Europe. You can look them up. Bornean Orangutan Survival Foundation. Boss F is, and we work together with them as well at the station. Conservation work, like the alternative livelihoods with the local people, reforestation projects, and we do more of the research. But we've worked together on different programs with environmental education. And of course, with the communities, we provide alternative livelihoods through the project for about 20 different households in the area. So, and now we're expanding our project to other primate species and insects and all different. So we've been hiring a lot more assistants lately, which has been great. I feel very lucky that I have such strong support for the work that we've done the past 20 years. The government's been really supportive as well of the work we do. They come like every once in a while and visits us. It's really nice.
Question from our Zoom audience. They're asking if a student wanted to get started in this field, what should they do?
So if a student wants to get interested in, if they're interested in doing primatology, or doing tropical research, my advice would always be to try to do, if you're an undergrad, a study abroad program. There are a lot of field-based study abroad programs, and often there's funding to students through universities where they are, or through the agencies themselves. For example, the Organization for Tropical Studies runs several programs. There are quite a few field schools for primatology, like this down in Costa Rica. We run one through Rutgers. So next summer, I'll be running a study abroad program. I can send Dr. Travis the flyer universities to come on our study abroad programs. And that's a 6-week program, or maybe it's 4 weeks now, a 4-week program in Indonesia. And we spend 10 days at the field site, and then we go to a marine ecosystem, and we do coral reef restoration and learn about sea turtle release programs, mangrove restoration. And then we also do a visit in Yogyakarta, which is kind of the cultural capital of Indonesia, where they have some beautiful temples that we go visit and learn about local culture there. So it's a really fun program, and I'll be running it next summer. I run it every other year, so I'm not running it this summer, but I'll run it next summer. So I'll send the flyer for sure, because we would love, we often have a lot of undergrads from other universities attend the course, and you can get, it's actually 2 courses. So it's a 6-credit month-long program. Yes.
So this takes a really long time to gather all the evidence and stuff. So what's the average, I guess, time for you to publish it?
So the question is, what's the average timeline for me to publish a paper once I gather my data? You do it a lot faster. Unfortunately, I've got a lot of administrative duties now as grad program director, and I'm directing the center, and the grad program director position is 6 years, and it's going to end soon. So I'm kind of ready. These data are collected now for 10 years. We can actually publish it. So I'm almost done with the manuscript for this one. But then there's been other papers that we've done that have been a much, a little bit of a shorter time frame. So usually I like to have at least 3 years of data, just because orangutans are more solitary. So our sample sizes tend to be smaller than you would get in like more social living primates. Yes.
You mentioned that sounds like you're looking to work on other primates in the same locality. And do you have any information on, because it sounds like orangutans are kind of unique in at least some of this physiology. Is there any evidence that that the other animals who are living in the same area, dealing with the same fluctuations in fruit availability, have different strategies for dealing with this, or are they just?
So we just. We see very similar patterns, how much ecology is influencing the physiology and the gut microbiome that we observe at these 2 different sites. And so we're adding gibbons and we're adding macaques to our study. And gibbons, actually, we've done some studies with my Indonesian colleagues on gibbons, and we found that they use a very different strategy. They just travel really far. They actually increase travel during the low fruit period, whereas orangutans decrease it, because they are really fruit specialists. So they're moving from fruit tree to fruit tree, but they also can move a lot faster. So it'll be interesting to see their physiology and how that varies as well. And the macaques, they're more omnivorous, so we'll see what they're doing.
I want to play off something Dr. Holbrook said a little bit ago of, he had mentioned the palm oil, and you had said that the orangutans do really well in the wild, especially compared to getting fat in the zoos. But they're losing habitat in different places. And so I guess they're doing well where there's full structural habitat, and then what's happening in the places where they're experiencing different versions of habitat loss?
That's a really great question. So, yeah, so we're in a protected area, somewhat protected. I mean, there's still logging and hunting that goes on, but they're relatively protected. But some studies have shown that orangutans can do really well in kind of marginal secondary forests as well. It's just in these palm oil plantations where they don't do as well, and they're often hunted as well because they eat the crops. They definitely raid crops. So the government has gotten a lot in Indonesia, has gotten a lot better in terms of, protecting the protected areas and not granting as many palm oil concessions. So a lot of the concessions that have been granted are on previously logged land. And I think that there's definitely more of an initiative now to conserve the habitats where they are. In Sumatra, it's a little more dire in terms of conversion of forests to hydroelectric dams and to plantations. So it'll be interesting. In the Sumatran orangutans, there's only about 4,500 left, whereas in Borneo, there's about 45,000 to 50,000 left. So Sumatra is definitely in a much more dire state than Borneo right now. But we're hoping that by, perhaps working with the local government on ecotourism projects and thinking of creative ways to increase kind of the income for the country by using the orangutans as kind of umbrella species, that we hope that will help. So we've been working closely with our Indonesian colleagues on that as well. You're welcome. Good question. And we really don't know. So colleague, Jessica Rothman, that did the work on them and found that they're kind of really overshooting their protein to maintain energy intake. We don't know what, if even if they do have adaptations, we haven't collected their urine to look at urea concentrations or delta N15 or urinary C peptides. So that's a really interesting question. Like, how do they cope with all that excess protein? Don't know. Yeah. And maybe they're not, maybe a lot of it is bound to fiber as well. It could be that they're also getting a lot of tannins and they're getting a lot of fiber because they're eating a lot of leaves. So a lot of that protein is also bound. It might not be, yeah, it may not be converted.
I think we have time for one more question. Oh, great.
So you said that they're fairly solitary in adulthood, at least the males, and they think you're crazy. Do they ever show any signs of aggression towards humans invading their space, or at least in your experience, or are they just like, what are these weird things doing down?
Yeah, I mean, we've had experiences where, so one of the things that the big phalange males do, like Nico here with the big facial discs, they do when they get when they do this to other males too, because they're violent with other phalange males. They'll push trees over. So like they'll find a tree that's decaying or rotting and they'll just like, somehow they're able to like push it over. So they will try to get us that way. If they're really trying to get us, or if they're display, because it's also a very loud audible display. They do these long call vocalizations, but the tree crash is like an actual display and it makes you can hear it from really far away, almost a kilometer away. You can hear it. I've had them come at me and they do like, so if we get too close to them, the males, we get too close to them and they're like, if we've got our cameras and we're getting too close, they'll come down really fast at you. And if you run from them, they'll chase you. So you have to hold your ground and just look away. Like, it's like a dominance thing. So if they know that you're submissive, so I usually, when they come at me, I'll just like kind of turn away and not look them in the eye. And then they'll usually just like breathe on you and then walk away. But they've never like attacked us or done any of that. It's just scary, because they're big. The females don't usually, they don't come down to us. They let it when they don't want you, when they want to come down to the ground, like if they want to drink water, or maybe there's a termite nest or something that they want to get to and you're in the way, they'll do a vocalization. It's called a kiss squeak, where they like go like that. And that means like, get away, like you're too close. It's a warning. So then we'll like back up and then they'll come down. So yeah, but they're really, the wild ones don't, I mean, ones in rehabilitation centers will come after humans, but the wild ones won't. The males will show their dominance for sure. But we you try not, if you don't run, they're not going to chase after you. If you run, they'll chase after you. So you just have to like hold your ground. It's hard though, because you're like scared, they're like, 200 something, 300 pounds, and they're coming at you with these big teeth. So they're strong. I mean, they can rip your head off. So, but they don't. So, yeah.
All right. Yeah.
Oh yeah, come work with me. No, they're a lot of fun too. They're beautiful. They're so cool. They're very cool animals. So,
All right, let's thank Dr. Rowe one more time.
thank you. Thank you.